So welcome everyone. It's a pleasure to have you all here for this part of the summer school, the first session, which is on machine braining, machine learning tools for understanding brain function. So let me just introduce the speakers first. So Andrea graduated in physics in 2016 and obtained a master's degree in computer science in 2017 with a specialization in machine learning and neuroinformatics. She's currently doing her PhD thesis. So am I. So chocolas. Chocolas is like, we're in the same boat. In the RIIAA lab with a focus in understanding the dynamics of hippocampal oscillations. Our second tallerista will be Pablo Varona, who's a professor in the group of neurocomputation in Madrid, in la Universidad Autónoma de Madrid. His group is called Neurocomputación Biológica. He has registered several patents and has been author of several publications in journals of international caliber. I guess that will be in Spanish, I'm not sure. Kemere, he got his PhD, he's the third speaker. His PhD from the Stanford University in the electrical engineering department or school. He was a postdoc in UCSF at the Neuroscience Center before joining the faculty of RIIAA in 2011. Kemere has won an NSF prizes in 2013 and 2014, has received multiple scholarships, including some from the National Science Foundation to study cognitive systems and other National Science Foundation prizes to study how memory is stored in the brain. And our fourth speaker is Dr. Vitor, who graduated from the Electrical Engineering and Informatics School in 2011, obtaining his PhD in neuroscience in el Instituto del Cerebro de la Universidad Federal de Rio Grande from Brazil. In 2012, Vitor received a scholarship from Ciencias Sin Fronteras from the Ministry of Education in Brazil to spend 1 year in the Leicester University, where he developed new methods to extract brain activity information. And he currently works as a researcher in the University of Oxford at the dynamic unit of brain networks. So it's a pleasure to have you all here, and I'll leave you the floor. Let's hope we don't have any trouble sharing screens, but I guess that should be fine. So I guess you can go first, Andrea.
Okay, actually the order is going to be a little bit different.
Okay.
I can just share the screen with the new order. Let me check.
Yeah, that's perfect. I'm just gonna be here in case there's any technical problem, but.
Okay. So yeah, that's it. I think everyone's looking at it. Yeah, so well, thank you very much. We want to thank RIIAA for hosting this workshop. The order is basically, instead of being me the first, I'm going to be the last one, and the first one speaking is going to be Dr. Pablo Varona, talking about artificial neural networks, machine learning, and related AI paradigms in neuroscience. Then Dr. Kemere, using hidden Markov models to unveil neural structure. Following with Dr. Vitor Lopes-dos-Santos, analyzing LFP spectral patterns with wavelets and ICA. And finally, me with convolutional neural networks to explore electrophysiological events. And each of these talks is going to last about 20 minutes, and there's going to be like 10 minutes of questions or discussion. So if anyone during the talk has a question, you can maybe post it in the chat of Zoom, and then at the end of the talk, it depends, we can talk it out loud, you can say it or I can read it out loud, it depends on how many questions. So, okay, thank you very much. We can start with Pablo.
Can you hear me?
Yeah.
Okay, so let me see if you can access my screen.
Yeah, yeah, we can see it.
hope that you are seeing the right screen. I have to.
Yes, the actual presentation. We were seeing it.
Notice that the sharing of the screen has stopped because the screen is connected.
However, I cannot hear you very well. If you could, yeah, get closer to the microphone.
Can you hear me now?
Yeah, a bit better.
If you want, I can try to increase my volume. Otherwise, okay, let me start, and if you find any problems in the sound or in the screen, please let me know. So first of all, thanks a lot for the invitation. Unique opportunity to communicate several, an introductory lesson regarding the use of artificial neural networks, machine learning, and other artificial intelligence paradigms in neuroscience. But of course, this is a very broad subject, and in 20 minutes, I can only be very brief. So that's why I prepare a lot of bibliography for you. And I just want to emphasize a few of the advantages of using artificial intelligence in neuroscience research, and also how artificial intelligence can be related to neuroscience bidirectionally, and point out the main problems and also some easy solutions for some of these problems. So let's start by saying that modern neuroscience is multidisciplinary. Actually,
excuse me, Pablo. I cannot see your screen.
We were able to see it earlier.
Yeah, and then you unshared it, I think.
Again.
We could see it before. I don't know, like, yeah, when you were asking if we see your screen.
Right now?
No. Now. Yes. Now. Thank you. Sorry.
Let me give. So you can see it now, right? Because I changed it slightly. Okay, good.
Yeah, but we are seeing like now. Yeah, now it's okay. Thank you. Yeah.
Okay, so I was saying that modern science is multidisciplinary, and this goes for any science. This goes for physics, biology, engineering. And of course, when we are talking about neuroscience, of course, neuroscience is multidisciplinary. We use current knowledge from biology, from medicine, from psychology, but also from engineering, many kinds of engineering, prosthetic engineering, brain-computer interface engineering, and also mathematics, of course, physics, chemistry, etc. Neuroscience, in fact, has always been interdisciplinary. And here you have a few examples just to name relatively old or relatively recent examples. We can focus on McCulloch and Pitts, who designed for the first what is considered the first artificial neural network. And these two persons, one of them was just a neurophysiologist, and the other was a self-taught logician. And Walter Pitts was actually a homeless who taught himself everything relating mathematics and logics, and a little bit of neuroscience. And he went to live to McCulloch's house, and together they create a very simplified model of a neural network. At the beginning, they were not thinking about designing an artificial neural network. They were trying to explain neural behavior. But with their simplifications, they could formalize the first artificial neural network. And their formalism was very simple. So neurons were connected to each other. The connections would be expressed as weights, and the neurons just gather the inputs, the weighted inputs, and through an activation function, created a response. And this was the basic artificial the first artificial neural network that was created. And a little bit later, just a few years later, Donald Hebb, who was a psychologist, formalized the first learning rule. He just proposed that when a neuron repeatedly fires because of some other neuron fire, then the strength of the connection increased. And this paradigm, which is called the Hebb learning rule, has been used extensively, not only to explain neuroscience research, but also to develop artificial neural networks. So seems in my screen also a little bit of, let me see if I can get rid of this. I don't know if you can see this. Can you hear me at all?
Yes, yes.
Okay.
We are seeing now joint neuroscience.
For some reason, I have some part of the in my screen, and I don't know if you are seeing that, and this is hidden the titles of my slides. Is that so?
No, joint neuroscience and AI efforts.
Okay. So it's in the middle. Okay. So there is a long tradition of joint efforts in neuroscience and artificial intelligence. And while I'm not going to review all of them, I just want to emphasize a few examples. For example, Stephen Grossberg is a well-known scientist in artificial intelligence. He has developed the adaptive resonance theory, which is not only an artificial neural network, a type of artificial neural network, it's also a paradigm, a theoretical paradigm to understand brain function altogether. So it intends to solve different problems, both in the context of artificial intelligence and also in the context of neuroscience. Also, John Hopfield, he created his associative network in the early 80s. And this has been a paradigm that has also been used to understand associative memory in neuroscience, both in neuroscience research and also in applied artificial networks, artificial neural network studies. Terry-Seniorsky also has proposed many different types of learning paradigms. He's the co-author of World-Match Machines and also the RIIAA technique in machine learning. And he's also a very good example of joint work, both in neuroscience and in artificial intelligence. And finally, we can also name Carl Friston, who is the author of the proposal of the free energy principle as a way to explain the Bayesian brain, which is also a very useful concept in artificial intelligence. So, but let's recall a few facts about the brain in order to assess how artificial intelligence can help neuroscience. If we estimate the number of neurons in the brain, we can say that the number of neurons is around 10 to the power of 11. This is a huge number of neurons. And the number of synapses, considering that on average, the neurons, each neuron is connected in the human brain to 1,000 neurons, we have 10 to the power of 14. It's even larger number of synapses in our brain, an incredible large number. And of course, the full connectivity information does not fit in our connectome. The connectivity in our brain is shaped because of development, because of behavior, because of the interaction. So this hints that we need some help to understand brain activity, not only because we have very large numbers, we have a very large dimensional space, but also because the brain is only partially observable. What does this mean? It means that we can only record a few variables involved in the overall information processing, for example, voltage, intercellular calcium, volt signal, etc. In addition to that, neural dynamics is highly non-stationary and also adaptive, mostly working in transient regime. There is no time to reach, in many cases, because the information is flowing constantly. There is no time to reach a steady state. Neural net systems work in multiple dimensions, not only at the subcellular level, but also at the cellular level between different subcircuits, small circuits, large circuits, whole systems, and so on. And also in different time scales, from a few milliseconds all the way to hours or even days. There are also multiple code transformations. There is not a single code in neural information processing. The codes are changing at the different sensory motor transformations. The activity is history dependent, and the processing is also closed loop. So this is why studying problems, any kind of problem in neuroscience, is usually very hard. In addition to that, I want to emphasize that most neural activity is transient and sequential. So the subcellular biochemical processes occur always in a sequential manner. The different channels open and close in a sequential manner to shape the membrane potentials. The switching dynamics among neurons in small circuits is also both sequential and transient. And there are many types of interactions that are inhibitory and excitatory in a closed loop. So these are all examples, including examples from imaging, from fMRI, in which you can see that the activity is always sequential and in most cases also transient. So it is very hard to study different problems in neuroscience, taking all this into account. And this is where artificial intelligence can become handy. So how can artificial intelligence help in all these problems? First of all, we can have novel analytical tools that can deal with the partial observation nature of neuroscience research. Also, they can deal with the sequential dynamics, with the different multi-interacting spatial and temporal scales in the brain. We can also use artificial intelligence to build models and theoretical formalisms that can describe this kind of dynamics and also their associated information processing. And finally, we can also use artificial intelligence to design novel experimental protocols that use both novel analytical tools, in some cases in real time, as we are performing our experiment, and second, theoretical formalisms that are in direct contact with experimental data. And in some cases, they are driven the simulation protocols. Okay, so machine learning tools provide automatic data association, classification, clustering, and prediction. It is very important to understand these different types of solutions that machine learning provides. And I'm going to give just a very few examples for you to get this very basic knowledge. We can also use genetic algorithms and evolutionary strategies in order to estimate parameters in computational neuroscience, in order to build models, which typically, if they are realistic, they have many, many parameters. We can also use things like a fuzzy logic from artificial intelligence to build activity-dependent stimulation and closed loops, in particular for online decision-making. And also we have a myriad of tools for neuroscience from artificial intelligence related to applications in the context of biomedical engineering and biomedical applications in general. So let's start with some very basic understanding of artificial intelligence and in particular machine learning. What are learning machines? Learning machines are just mathematical input-output models or algorithms or processes that lend themselves to some kind of parametrization to achieve a given goal. In particular, most times we want to associate some input to some output. And how do they learn? They learn by adjusting the machine parameters. If we are talking about an artificial neural network, we are just adjusting the weights of the connections until our goal is reached. And typically the goal in machine learning paradigms is to minimize an error measure. It can be any kind of error, any kind of loss function. For example, the mean square error between the input and the output. The machine learning then tries to find a specific algorithm in order to have a parametricized input-output transformation that will minimize this error, this performance measure of what the machine wants to achieve. And in this context, we have different types of learnings. We have supervised learning. In this kind of problems, both the inputs and the outputs are known in some examples, and we want that the machine can generalize. That is, given any input that the machine has not seen before, will lead to the right output if the machine is working fine, if we have trained the machine correctly. So in supervised learning, the targets are known, and the model estimates or predicts these outputs or converges to the outputs. So these known targets guide or supervise the learning process. And once the machine is trained, then we can apply it to any input data, and it doesn't, we don't need to have a desired output. Then this is the task of the machine. The machine will predict or provide the output. In unsupervised learning, there are no supervising outputs. And this is a common case in neuroscience. We have just inputs, and we want that the machine learns some association or finds some structure in the data. And we can use this kind of unsupervised paradigms, for example, clustering. It is often a first step towards supervised learning, and many unsupervised paradigms are used to establish the weights of the connections of artificial neural networks that are at the end supervised. And then we have also semi-supervised approaches in which some patterns have targets associated and others do not. And also this is a very common case in neuroscience, because we can only tag some of the patterns, and then other patterns are not tagged, they don't have outputs associated. And then we are in this situation of a semi-supervised case of learning. Okay, so this was just some very basics about machine learning. But what we want to discuss is how can artificial intelligence help to neuroscience. And one of the themes in which artificial intelligence can help is in assessing morphology, in finding neuron or synapse types in an even circuitry. We can also assess physiology. We can study neural dynamics in relationship to information processing. And information processing, of course, is information encoding, decoding, coordination, information execution, and also, very importantly, information creation, because the brain creates information. It doesn't only transduce information. So we have morphology, physiology. We can also assess models. Typically, in computational neuroscience, we have models with huge parameter spaces. And these parameter spaces can not always be constrained by the data that we have. And we need tools from artificial intelligence to give values to the hundreds, thousands, or even millions of parameters that our models may have. And also, we can use artificial intelligence for a closer and more realistic interaction with the nervous system, using tools also from artificial intelligence and devices that are designed together with artificial intelligence paradigms. For example, implants, brain-computer interfaces, rehabilitation robotics, and so on. So let's go step by step with just some examples. I can only give you in the time that I have a few references for each of these cases. Assessing morphology, we typically want to automatically discover neuron types, find neurons under the microscope, and also characterize circuitry. And we can use machine learning methods for that. We can use artificial neural networks. We can use many kinds of statistically based artificial intelligence paradigms for assessing morphology. We can also use theoretical formalisms from network science that is used in many other disciplines like social networks, in order to assess also connectivity and circuitry in the brain. Now, of course, tools from graph theory. Regarding physiological studies, of course, this is one of the hardest problems in neuroscience research, right? To assess physiology in the context that I have given you before, right? The brain is only partially observable. So that's why we need automatic discovery and classification of neural activity. We want to find and classify spikes, bursts, event-related potentials, and also complex sequential activations between different systems. And we want to do it as automatically as possible, right? And always with our mind in relating all these events with neural function. And of course, it's a hard task because neural activity is not 100% reproducible. You can have here an example of different spikes of exactly the same neuron in time. They are overlapped in time. As you can see, they are not exactly the same. They vary in time. And the same goes for event-related potentials like P100, which are used in brain-computer interfaces. So in general, also, we want to implement novel experimental protocols, model-driven stimulation, or interaction that relies on finding events, in many cases in real time. So you have many examples of different techniques that can do this, but basically we can classify neural data and neural physiology using these tools. This has a lot of problems, and I will mention these problems, and I will mention some of the solutions a little bit later. Of course, when we are in the context of computational neuroscience, we already said that a big problem is to fit the huge parameter space that these models may have. The more realistic the model, the larger the size of its parameter space. And many artificial intelligence paradigms, and in particular evolutionary strategies, and also genetic algorithms, are handy to assess this parameter space. We have to understand that this is also a very difficult task. And when we have a model with many parameters, we are prone to have many problems, even if we have a good method to constrain the parameter space. And I will mention some of these problems in a few minutes. We can also use artificial intelligence to promote more realistic interaction and more effective interaction with the nervous system. And this is the case when we're talking about implants, brain-computer interfaces, and rehabilitation robotics. And in these references, you can find some of these solutions, some of these representative solutions. This reference list is not complete. It's just some of the references that I like it, but they are not complete. You have many different examples of all these cases. But then let's go to some of the problems in each of these cases. When we are using artificial intelligence to assess morphology and physiology, we should be aware that in many cases we have only small training sets with unrepresented classes. If we want to classify spikes or bursts or event-related potentials, any kind of physiological activity, of course, we are restricted to the amount of data that we have. And also, if this data comes from different days or from different setups, we have the problem of variability, not only because intrinsic variability of the nervous system, but because the different variability among our devices that we use to record neural data. Also, in many cases, we use manual tagging for targets. In the supervised learning approach, we need both inputs and known targets, right? And if we tag the targets manually, then typically this means that they are not very reliable or not 100% reliable, and that we have very small amount of tags, right? And then, of course, in the context of machine learning, we are always facing the problem of overfitting. The problem of overfitting is illustrated here. We can just see how the error evolves as our machine learning paradigm, for example, an artificial neural network is learning with regard to the number of epochs, right? So this is the loss function, the error is being reduced as we progress with the epochs in the learning, as we have presented our patterns to the machine learning paradigm. But if at the same time we calculate the error, the loss of patterns that we didn't show during the learning, we can see that sometimes the learning stops earlier than with the patterns that we are using for the learning process. And this means that at this point, we are overfitting our data. The machine learning is learning too well the patterns that we are using as examples, and it's losing the capability to generalize. And well, many examples in the neuroscience literature that uses artificial neural networks to fit parameters have overfitting very clearly in their data. So how can we solve these problems? We can have synthetic data and semi-supervised learning. From the data that we have, we can generate new data using the models, for example, or using different statistical techniques to introduce variability in the data, variability that can be biologically justified. We can also use semi-supervised approaches to solve the problem of having small training sets. We can also have artificial intelligence helping us with automatic tagging. This means that we want to identify events by using functional, other functional events that we can record at the same time of events that we are interested to classify. So this, of course, means having harder experiments to design, but we can use this technique of automatic tagging in order to avoid the problems of manual tagging or unreliable tagging. We can also use regularization techniques, for example, if we are using deep neural networks, to deal with the overfitting, and we can always check whether our learning overfits or not by having this very simple analysis when we train our machine learning paradigms. Okay, we can also, regarding model building, we already mentioned that typically we have a very huge parameter space. And also, in many cases, we have low reproducibility of experimental observations under different conditions. Our model sometimes can reproduce the output to a given input, and we have a very different input or very different, we are modeling a very different experimental condition. In many cases, our model needs to be reshaped, or we need to find novel parameters in our model in order to reproduce this second condition. Right? In some cases, we have stability issues. Neurons are very robust, and we can do all kinds of nasty things to them, introducing currents, introducing different drugs, and they always oscillate, they always produce activity, right? But models, it's a different way, it's a different game, right? We sometimes, when we have different inputs, the model just completely behaves in a very different manner, right? So we can provide some solutions to these problems by using genetic algorithms and novel experimental paradigms that relate the models to the data that we are acquiring. We can also use dynamical analysis to assess low reproducibility of experimental observations. And also we can use theoretical frameworks to drive the model building. When we are building a model, we need to think, okay, not just start by putting things together in a bottom-up approach. We can use a simultaneous top-down and bottom-up model approaches that rely on our knowledge of dynamical analysis and also on our knowledge of multi-compartmental modeling, for example. Regarding the closed-loop stimulation, okay, we need to detect events and produce output in real time. This is the case in brain-machine interfaces, in many rehabilitation simulation protocols. We really need to detect events and produce a stimulus in real time. And of course, closed-loop experiments are harder to design than traditional stimulus response experiments in neuroscience research. So they are hard to design, and that's why we need some help from artificial intelligence for that. And finally, we typically have a huge amount of data to analyze offline. So how can we solve these problems? Well, for the first problem, we can use high-performance artificial intelligence and machines, in particular the use of GPUs, to have different algorithms that are working to detect events, and they can use artificial intelligence paradigms for this detection and for this classification. Sometimes we not only want to detect events, we also want to classify them, or we want to predict them, right? And of course, we need that these algorithms work fast. We can have many artificial intelligence paradigms that are efficient, but they don't work fast enough. And in order to do that, we will need to select some of those algorithms that will work in real time. And we want to implement them in hardware architectures that will run them very fast. We can also use real-time software, because sometimes the issue is not the fast performance, but the timing, the accurate timing of our simulation. And for that, we need real-time software. For problem 2, hard to design experiments, we can always use a goal-driven approach, having goal-driven performance measures. We need to be very aware of what is our goal in the closed loop experiment. And with that goal in mind, we can design different measures to assess the performance, and we can explore our parameters during the simulation in real time using many kinds of machine learning paradigms. And we can use hybrid circuits. Hybrid circuits are built with artificial paradigms, artificial models, for example, artificial neurons or artificial circuits working together with living circuits. And finally, we can rely on online analysis. So if we design all these event detection tools and internal representation tools with artificial intelligence, in many cases, we can rely just on the online analysis. We don't need to do the offline analysis. But okay, let's go back to the beginning. I'm just ending my talk now. Modern neuroscience is multidisciplinary, and in particular, neuroscience is interdisciplinary. And the same way that artificial intelligence can help neuroscience research can help the development of novel artificial intelligence. And we already mentioned the example of McCulloch and Pitts. At their time, they didn't distinguish too much what was artificial intelligence, because for the very simple reason that this concept was not even created at that time, right? And neuroscience, they were always working together with theoretical and with applied concepts, and also with wet concepts. So if we take a look at artificial neural networks, we just can realize that the bioinspiration in artificial neural networks, even in modern artificial neural networks, goes back to the 1940s of the last century. So the basic units are of processing information are the neurons. They are connected among themselves. They decide an output. The output is transmitted to other neurons. The network learns because we change the connections. And in many cases, we just use the Hebb rule. And in other modern artificial intelligence paradigms, we use other types of rules that minimize our error function, as we mentioned before. Right? But there has been a huge amount of progress since the 1940s. And you can see, for example, that there are 30,000 papers in the Journal of Neuroscience, just to name a representative journal, right? But we have many different journals, and the progress has been huge. So we can use many elements of bioinspiration in order to help artificial intelligence from neuroscience knowledge. We can design artificial neural networks, knowing the design of living neural networks. We can include different types of dynamics that are known in living neural networks, in particular the ability to process sequences in artificial paradigms. We can, of course, use not only the approach of the processing itself, but the materials that are involved in the information processing. We can control architectures and self-regulation. This is a big secret of biology, right? The self-regulation and the self-repair nature of the information processing. We can also get bioinspiration for the miniaturization. The size of the neurons are really very small, and they are very tough information processing devices. Neurons, as we mentioned before, create information. They not only transduce information. This is something that artificial intelligence paradigms don't do very well yet. There are many types of sensory systems, perception and communication mechanisms which bases are known and can be transferred to engineering. And of course, there are many different types of elements that can be used for bioinspiration in addition to those that I list here. In particular, energy efficiency, which is also a theme that is being overtaken now by many different hardware scientists. So I want to end that there is a huge amount of knowledge that can be transferred to artificial neural networks. Neuron diversity, information encoding, processing and execution diversity, the type of dynamics that involves sequential activity, interacting spatial and temporal scales. So not just one single scale, not just one single encoding mechanism, not just one single learning mechanism. Information processing is always history dependent, and this is something that has not been taken into account in many artificial intelligence paradigms. So let's finish with some conclusions. The first one is that artificial intelligence tools are always handy to deal with the complexity of problems at many description levels in neuroscience research, and also to assess overall brain function. That the second conclusion is that artificial intelligence is not a universal solution for neuroscience research. You need to be aware of the problems that we have discussed and also be aware of some possible solutions. And finally, that the knowledge transfer from neuroscience to artificial intelligence is also possible, and that nowadays they should work simultaneously, right? We can get bioinspiration from neuroscience research to design novel artificial intelligence paradigms and also robotics and brain-computer interfaces. And I just want to conclude with some additional references that I will leave while I answer to your questions. And here is my email address, just in case you have some questions beyond what we can discuss today. I think I already took a lot of time. Thank you.
Thank you very much, Pablo. It's been, as you said at the beginning, it's not easy to summarize like the interactions between neuroscience and artificial intelligence, but it's been a wonderful summary. Thank you very much. There has not been questions in the chat. Do any one of the attendants want to ask anything? I had a question, but I think, yeah, we are a bit of, I think we have to follow to the next talk, because it's, yeah, it's been, here, I think there's a question.
There's
Okay.
a question very quickly. The question is, when talking about model building, how do these simultaneous-program and bottom model building stories look like? Okay, this means that you can have a bottom-up approach, which is very typical in neuroscience research. We know that the neurons are made out of different compartments. We can characterize these compartments and have many different channels in all these compartments. At the end, we can reproduce some of the physiology of the neuron using all this information, all this bottom-up approach. But at the same time, we can use a top-down approach in which we have a theoretical formalism that says something about the physiology, about our goal-related physiology. For example, that we want that our neuron generates spike patterns that have some given sequence, and that when we build a network with these neurons, we will create the type of sequential activity that we observe in the physiology. So we can have a theoretical formalism that proposes how we can achieve these sequential activations, realistic sequential activations. And this theoretical formalism can be far from the different ways that you can use to implement the individual neurons. Okay, I don't know if this answers your question. You can have theoretical formalisms that tell us more about the dynamics and less about the specific implementation of our models, our neuron and network models.
Okay, thank you very much. So I think we go on with Dr. Kemere.
We could see your screen, but you're muted, I think. Or very low volume.
Yeah, I cannot hear you either.
How about now?
Yes.
Pretty clear?
Yes.
Okay.
Thank you.
Okay.
We're not seeing your screen right now, though. Anymore.
Yes, no, I know that. Wonderful. Thank you so much for having me. And I give a special greeting to the students at UNAM who are attending. I think I've seen a couple in the chat. I have a first year PhD student who just arrived here in Houston, who is an alum and alumna, and she told me about the challenges that you guys have had this semester. And so congratulations on having made it through the summer and to a new fall. Today I'm going to talk about work that was, or largely work that was published in eLife in 2018. So if you're curious about the implementations, or if you have questions, feel free to email me. Or also you can see we have a GitHub page that has the code for all of the figures in that paper. And I'm happy also to respond to issues if there's problems running that code. And the work was done largely by two intrepid graduate students, Karush Mabodi and Etienne Ackerman. Karush is just finishing his PhD now at the University of Michigan with Cameron Diba, who's my close collaborator. And Etienne now is working for Uber. It just sort of shows how you can work in the field of machine learning and neuroscience and then find that the skills that you've learned have application in many broader areas. And thank you also to the funders who have supported this work. As a little bit of background, I very much like the word latent variable. And so I don't know of those of you who are in the audience now or those who will be in the future, whether that is a familiar word or not. But basically the idea is that when you observe something in the world, that there may be something that you don't observe that explains it. Interest that word explain is very popular these days in the context of deep learning. So a big question is, can we have a black box deep learning model that produces some amazing performance, but we recognize that it does that because it takes some sort of shortcut. And if we can understand how it is taking those shortcuts to achieve its success in either classification or identification or control, then that will help us understand whether it will work in new contexts and understand how to work with it. Interestingly, that same idea, so we have explainable artificial intelligence, but we actually are also very interested in explainability in neuroscience, but we use very neuroscience specific terms like a stimulus response or a tuning curve that says that we understand how neurons in a particular part of the brain may represent their perceptions of the brain's perceptions of the world, or perhaps how they represent or control the activity of the body. So machine learning is a very valuable tool in the specific context of explainability, because it can help us automate discovering these representations, so the variables that explain why the neurons are acting the way that they do. And there's a really nice review from 2014 in which the authors identified sort of three basic categories of ways that this is useful. So one is just to visualize the latent variables, or the spaces, the dimensions of the representation that the neurons are sort of encoding as a large group. Then that way, if we can see these lower dimensional spaces that describe how the neurons are acting, then we'll understand what those spaces are or what the neurons are doing. More broadly, they can help us understand perhaps how population responses, so the activity of many neurons, in response to a stimulus or a controlling an action are changing based on different stimuli. And then also, the tools are useful because when we have this latent variable or this thing that is explaining why our neurons are acting the way they are, oftentimes for brain-machine interface, we might want to decode that. We might want to know what the low-dimensional thing is as opposed to just the raw neural activity that we're seeing. I am a very big fan of a technique called probabilistic generative models. So this is a subset of machine learning where our conceptual framework is that there's, can you see my mouse cursor moving? Yes, okay, great. Yeah. So the conceptual framework is that there's some latent variable or a state and that we can't observe, and that we can define a probabilistic relationship from that state to the thing that we do observe. And so I've written these, this is like LaTeX font, right? So I've written these as bold characters to represent the fact that both our observation might be high dimensional, our state might be high dimensional. And the probabilistic generative model approach is very nice because oftentimes if we can draw a model of relationships and we can define the probabilities in some admittedly small subset of probability distributions, then we can just write down solutions that will allow us to decode, estimate the states, for example, learn model parameters, or visualize things so invert, so go from data to latent variable. Now, I've drawn sort of this is called a graphical model that describes this probabilistic generative model. I've drawn sort of three more complex types of graphical models or probabilistic generative models. So the first is one in which we have our data that sort of exists independently from one time point to another. I think there's going to be another talk in this session that talks about general solutions for this problem of trying to define some relationship like this and then learn how the neural activity and the lower dimensional thing are related. We could generally call that dimensionality reduction. We can add in now the concept that we have a relationship that constrains how the latent variable, the low dimensional thing that describes our data, is evolving across time. Then we have a dynamical model. So now we've added another variable, and I've drawn this. For those of you who are experts, you'll know that this is representing a Markovian assumption. You don't have to worry about that if you're not an expert. But then we can also have things that are more complex. So we have a bioRxiv paper where we presented a model where now we instead of thinking that we could observe the activity of individual neurons, we think, okay, now we have some noisy representation of the activity of those neurons. So now we have a level of indirection here between our state, which is projecting to neurons, which is projecting to marks and the things that we're actually observing. And basically, the more blocks that you added, the more complexity that you add to one of these models, the harder it is to deal with. Also, the more complex these relationships are, the harder your model is. So I have here, this is a slide that I got from John Cunningham. He said that I should make sure to give credit also to Jonathan Pillow and Scott Linderman. That they've been basically presenting this slide at various conferences for a while. So we can think about for this class of models where there are dynamics, we can have dynamics that have a variety of relationships, from discrete to continuous, nonlinear, complex. And our observations can be as simple as sort of continuous, linear, high-dimensional Gaussian random variables to highly nonlinear things that can't even really be described with a simple probability distribution. And this is an area of active research in our field, in the field of machine learning, and also the field of what we might call neural machine learning. But the thing that I'm going to talk to you about today is this thing that's up here in the top, the old part of this category, which is a hidden Markov model. A hidden Markov model, these states are now not real valued, the states are discrete. there's one of k possible states that our latent variables evolving from. And the data that I'm going to talk about is the firing of individual neurons, which we'll model as a Poisson random variable. All right, so I want to show you how we have been using hidden Markov models to try to do unsupervised discovery of the tuning of neurons that we know are producing patterns that are related to memory. And then I want to show how we use the structure, so the probabilistic structure of the hidden Markov model as a way, as we leverage that as a way to get more insight into what we're seeing. Okay, so brief background for the non-neuroscientists in the audience. The hippocampus is a structure in the mammalian brain. There's a beautiful, I think someone at the University of Montana has a beautiful archive of histology from maybe 40 or 50 different mammalian species. And it's remarkable. So if you're a little bit of a neuroscience nerd, you may search for this. And what you'll see is that this structure, the hippocampus, is found in everything from aardvarks to polar bears to zebras. And it has the same structure, and you can see it instantly if you're looking at a histological slide. So it's very neat in that sense that it's preserved from what we might call more simple brains in the rodent all the way to the human. It's critical, we know now, from about almost 100 years of study for the formation of new memories. And there's some evidence that it also is critical for the recall of particularly detailed memories like that we would call episodic. So for example, if you remember a particular thing a particular event that happened to you, like in my case, I can remember learning to drive when I was 16 and being excited because I learned, this is in the old days where there were manual transmissions, I learned how to drop the clutch in my parents' Volkswagen. And I was at a red light and the light turned green and I dropped the clutch and then a car ran the light and T-boned us. And I remember a lot about that. It's very cold. Even though this is 30 years ago, that sort of memory with a detailed spatial relationships, temporal relationships, people, things requires the hippocampus. And on the science side, my lab is very interested in understanding how those memories are formed and recalled. And the rodent, if you stick an electrode into the hippocampus, you record the signals of the neurons there. And if you zoom out, so if you look at sort of slower time scales, you'll see something called the local field potential, which is a similar signal to what you would see to EEG electrodes that you put on the outside of the head, except it's more local to the general patterns that are happening in the hippocampus. In the local field potential, as animals are exploring an environment, in this case, this rat is running on a track. It's a linear track that may be receiving reward at either end. We see this oscillation in the local field potential, and the neurons, individual neurons that we can record with our electrodes, by and large exhibit a pattern called a place field. And this is a tuning curve. It's just the way that we explain what these neurons are doing is that we have discovered through experimental correlation that an individual neuron will be active in this particular part of the hippocampus when the animal is in a particular location. So for example, you'll see these 10 neurons as the animal runs from right to left. The neurons that correspond to the early part of the environment were active, and now the neurons that correspond to the later part of the environment are active. And I've drawn these little tick marks because the way that neurons signal with each other is through the release of neurotransmitter that happens in a burst called an action potential or a spike. So it's a discrete event that we can record with our extracellular electrodes. So this discovery of this particular tuning curve is now about 50 years old. It won the Nobel Prize in maybe 2013 or 16 or something like that. About 15 or 20 years later, it was noticed that during periods when animals were not moving in these mazes, that if you listened carefully to the spikes that you were seeing, that often during brief bursts, so now this is a time scale of seconds, this is a time scale of tens of milliseconds, these same neurons would be reactivated in the pattern and the sequence that they were activated while the animal was behaving. And this phenomenon was called replay. The idea is that the animal has experienced something, and then now during brief bursts, the hippocampus is recapitulating the pattern that it had when the animal was exploring. And the thought, now backed up by some evidence, is that this replay is one of the primary mechanisms that allows the hippocampus to teach the brain to learn, to save its memories. And there's some really nice experiments where we've detected the oscillations in the local field potential in real time and silenced the hippocampus, so blocked these things, and have shown that animals don't fail to learn without these brief periods. So we're interested in replay. Now, when we're thinking about hidden Markov models, the area CA1 of the hippocampus is very interesting. I just described it has different patterns in the local field potential, it has different patterns of spiking. And if you're familiar with hidden Markov models, they are often used to identify sequences of states. And so a very logical thing to do might be to say, I want to distinguish between the periods when the animal is in this state or mode of activity and this state or mode of activity, and I'm going to use a hidden Markov model to do that. We're not going to do that. Instead, we're going to look at states in a much finer time scale. In particular, we want to understand if we can use a hidden Markov model to represent or detect the activity that is happening during replay. So first, just quick background, how do we normally detect replay? So the process is pretty complicated. So the first thing is that we observe the animal as they run back and forth in the environment. Here's a given neuron. This gray trace is the animal's position in this 2-meter maze. So they're running back and forth, and there's this place cell. So this neuron that likes the particular locations in the maze. So from this activity, we would form a tuning curve, so a model of how the neuron responded when it was in when the animal was in different parts of the maze. We record many of such neurons, so now we have many tuning curves. Then we observe spiking during an event that we think might be a replay event. And we use a very simple probabilistic the simple Bayes rule inversion of taking this model, which gives us a firing rate or a Poisson rate, a probability of firing given position. We take our observations, so firing, and from them we invert that probability distribution, probability of firing given position to probability of position given many firing. And that gives us now a probability of the animal's location as a function of time. And so now here we have something which says during this brief 100 millisecond event, we decode that the animal imagined being in these periods. Now, for replay analysis, typically what we'll say is, well, our null hypothesis is that this activity was just random. And so to understand whether or not it was replay, we'll rely on the fact that the maze was a straight line. And so if the animal was remembering running in the maze, they should also be, these memories should happen in a straight line. So we do a lot of stuff. But the thing that's important is that we have this multi-step process that starts by observing the animal when they're running in the maze and observing the neural activity. Okay, so how are we going to use hidden Markov models to study these ripple or sharp wave ripple sequences? Let me see, is there a question in the chat? What non-mammals? Oh, there's also a question. So sorry, please do put questions in the chat. I love these questions. Reptiles and birds have an analog to the hippocampus. The hippocampus in mammals is curled underneath the cortex. The cortex is the outer sheet of the brain. The hippocampus sort of curls off of the end of it and up inside, and it has a very characteristic spiral shape. That's where the name hippocampus, or Latin for seahorse, comes from. In reptiles and birds, it doesn't curl as much. It's actually up on the surface, the analog. But there's some just in this past year, there's been some beautiful results showing that birds that cache seeds, so meaning that they gather seeds and then they bury them in the ground, have a this very similar place code in their analog of their hippocampus. And there's also evidence for these bursts, these ripple bursts, that I think in some sort of lizard called a dragon. I think there's a paper from a few years ago. There's even someone who has been so crazy as to try to record in the brain of a swimming fish, and they see somewhat similar patterns in a fish analog of this part of the brain. So this idea that you need, in order to form memories, that you need a specialized brain circuit seems to be well, okay, I think octopi, I do not believe have anything like a hippocampus. But apart from them, which are already a very, very neat class of like very other brains, apart from them, vertebrate memory seems to rely on having this specialized circuit. Before I go on, are there any other questions? And I realize for those of you who are future session organizers, the problem with giving tenured faculty 20 minutes is that they'll say, oh, I can present 30 slides in 20 minutes, no problem. And the reality is that they have not actually gotten any better at that in the 20 years that they've been faculty. And so I apologize. I will probably cut some stuff out at the end. But I would love to take questions during, so I'll keep an eye on the chat here. So please put more questions in the chat. Okay, so here's our data. Here's neurons. Here's period. This is the speed of the animal. And these blue bars, we've identified periods when the neurons are bursting in the way that we think they do when the rats are sitting still and having these potential replay periods. So the normal process, remember, starts with this activity and then decodes or builds a model to try to understand this activity. So instead, what we're going to do is we're going to start just with these bursts. And we're going to rely on the fact that a hidden Markov model can work in a mode that allows for unsupervised learning to see if we can learn the structure of the code for this environment just from these bursts. And I think this picture is important because it gives you a sense of the time scale. So there's a lot of activity when the animal's running. When they're sitting still, the hippocampus is largely quiet. There are periods when it becomes a little bit active. And then we have these bursts. These bursts are hundreds of milliseconds. This is many seconds. So we're saying, okay, we're going to take a much smaller amount of activity and see if the hippocampus, if we can learn about the hippocampus just by looking at that. Now these red dots have just been ordered by basically which one came first. So there's no so you see a sequence, but that's just because that's how the neurons fire. One neuron fires, then the next neuron fires. It doesn't actually necessarily imply anything interesting. Okay. For those of you who come from a machine learning background, I just wanted to very quickly throw out what a hidden Markov model is mathematically. So we describe this thing as a discrete probability. So if we have our states are defined by I and J, then the probability of transitioning from state I to state J is just a number. And those numbers have to add up to 1 in the right way. Our observation we're going to take is a vector of Poisson random variables. The Poisson distribution is the equivalent of the Gaussian in the sense that it represents the most generic or most random way that a random variable can produce integers. So 1 spike, 2 spikes, 10 spikes, as opposed to real value numbers. So we can draw these as pictures. So when we learn a hidden Markov model using the expectation maximization algorithm or the Baum-Welch algorithm or other approaches, we will learn a transition matrix, which we've drawn out here. And we will learn, because these are Poisson, they're described, each neuron will be described by one number, which is its firing rate, depending on the state, so conditioned on the state. So we'll learn a matrix also for our observation. So we have our state, our observation matrix. All right, so we take this data, we run the unsupervised learning, And then we can decode states. So now we can say what state were we in at each time bin. And great, we see some state sequences. And actually, okay, here's our state transition model and our firing rate model. And if you're like me and you really like machine learning tools, a lot of times you will spend a long time getting to this point. And then you'll say to yourself, okay, now what? I learned a model. I think it represents something, and it can be somewhat demoralizing. So I think for the majority of the talk, the rest of the talk, I'm going to talk about what to do when you've gotten to this point. And there's a hidden slide that I'm skipping over, but if you want to look through my slides later, I will share them with the organizers. We spent a long time just trying to characterize these things to say, are they real? Are they meaningful? The parameters that we've learned, do they mean something? But I realized that I think that in our case, the thing that really gave us confidence that we were learning something interesting was when we tried to think about how to relate it back to things in the real world that we had observed. And so the trick that we did was we took the model that we learned from these brief bursts and applied it to the neural activity that we observed when the animal was running around. And our hypothesis, our knowledge, our background knowledge is that the hippocampus is representing space in the world. And so we wanted to just validate that the thing that we were finding, the states that we were finding, corresponded to space in a nice way. And so what we could do is we could take the periods when the animal was running, decode the states. We know the position at those times, and we can just develop a correlation between state and position and normalize it in an obvious way. And if we do that, then what we find is that for our hidden Markov model, which we arbitrarily chose to have 30 states, that each of those states ends up being active or being most probable when the animal is in a different position on the track. And so we have some structure. And if we randomize our positions just as a simple control, so we'll take our neural activity during behavior and just randomize where the animal was corresponding to that neural activity, we get some, and then we do the same decoding process, then we get something that's very random. So this is actually real, right? It actually is the case that there seems to be a very nice correspondence between state and place. And so another way of saying that is the hidden Markov model has discovered, okay, if you go to machine learning conferences in the past maybe 5 years, I feel like the word manifold is something that people have liked very much. And it's a buzzword, but it's a very nice buzzword, so I'm going to use it. So we found that our states correspond to the manifold of space, one-dimensional space in this simple ways. Okay, and they may also represent other interesting things. And I'll try to come to that in the second part of the results here. So something interesting, here's the place fields of the neurons that we were decoding. If we do a simple place field analysis, here's these latent state fields that we've learned. It seems like that they span the environment a little bit more smoothly than the place cells do. Maybe that's interesting. But that answering the question, is that interesting, is something that always requires more effort. All right. So I've shown you that using no behavioral data, so just neural data, we've done unsupervised learning on seconds compared to tens of minutes. And we've learned that the hippocampus, during these brief bursts, that there's a manifold that corresponds to space in the neural activity. Okay, so I think for the sake of time, I'm going to skip this piece. I'll quickly show you that, or I'll go very quickly. We can use the probabilistic formation of the hidden Markov model to define a score. So now we can take a sequence and we can say, is this sequence likely given our model? And we can use that as a way of deciding whether or not something is actually replay. So now we can do cross-validation. So we train our model on some subset of data and ask whether a given event is consistent with that model that we've trained using this score from the statistical formation of the hidden Markov model. And we find that if we do that, that we end up with roughly the same performance of detecting replay as if we had used all of the behavioral data. So this red curve is the behavioral data. This blue curve is only the periods when the animal is bursting. Okay, so model congruence is a nice tool that leverages the hidden Markov model's statistical structure. All right, so the last thing I will show you, and I promise also this, I will try to do this somewhat quickly, is this question of, can we discover something with the hidden Markov model that, that we didn't know beforehand? And one of the things that we wanted to know whether we could discover is whether the animal is remembering multiple environments. And the hypothesis is that these replays correspond to the animal replaying. And sometimes we see bursts of activity that don't seem to be replayed. Could those be memories of another environment? And if that were the case, then we would expect that we should be able to see two discrete manifolds in our neural activity. So one manifold corresponding to the environment the animal has just experienced, and one manifold perhaps corresponding to another environment. And so we actually took some data where we had the animals exploring two environments in the same day, and we trained a model on the whole thing, and we found, okay, if we sorted our states, it looks like maybe there's two clusters here, but again, we already knew that there was two mazes being explored that day. So, okay, this is not, we haven't discovered anything. So then we came up with what I think is a neat idea, where we now wanted to leverage both what we knew about our data, so this idea that we know about place fields and we know about replay, and our hidden Markov model based replay analysis. And our idea was this. What we reasoned was, if there was a second manifold in the data, then if we enriched the data for that second manifold in our training data that we're using to learn our hidden Markov model, then we would see a lot of self-congruence in that firing. So we would see that the patterns were congruent with that enriched model. So how do we enrich a model to be representing what is not in the data? Well, the way that we can do that is we can take our place fields and learn the structure of the replay based on the place fields, and remove from our training data all of the events that correspond to good replay based on the place fields of the environment. And so this x-axis here represents what fraction of those events that we're removing. So this is removing very few events, this is removing very many events. And this y-axis corresponds to a measure that we came up with that basically says this is the self-congruence of the model. So if this number is high, then this is a model that represents something very nice. If this number is low, then basically it's just the model is just noise. And so what we see is that as we remove more and more events, these different data sets take different paths. And there's one in particular, this blue line, that seemed to take a path that was like a little bit, it didn't go down very quickly. We have a second thing here, which we just represent a second metric, which represents just the amount of data that we have, which is session quality, which is like the thickness of the lines. So this is actually a pretty poor quality session. But then, so it's decreasing, but it's actually starting from a very low level. But this one was a fairly high quality session. So we hypothesized that in this session, the animal was remembering something else that happened. We wanted to see if this approach would work. And so we pulled in some other data that we had where we knew that the animal was remembering, or we knew from a prior result, this is Carlson 2016, that the animal was remembering a different environment. And we found that in fact, when we took the same data in this environment, that this phenomenon of remote replay remembering something else was detectable this way. So we were excited. It seemed like we could leverage both our hidden Markov model, unsupervised learning-based approach, and our neuroscience knowledge of the data to discover something new, basically to discover when an animal was remembering multiple manifolds of the environment. And that actually is now the work that Kamardiba and I are still carrying out as part of an NIH-funded study. Okay, so I'll skip over this. All right, so we're very excited about hidden Markov models, ability to do unsupervised learning on neural activity. We used that as a way of providing a new technique for neural decoding, and we believe that it also may allow us to discover something neat about what happens during memory during offline periods. All right, so I'm sorry. I think that was 30 minutes. And so I think there's probably no time for questions. But I would love to take any quick ones now, and otherwise I'll just hand the baton over, and please feel free to email me questions. And Andrea, will you be collecting our slides?
Yeah, I think so. It's a good idea. Yeah, with all the references that you've been posting, I think it's. I had one question. Did you try to apply your model to data before the mice entering the maze? So what was happening before and the pre-play and all these things?
Yes. So for those of you who know about memory and memory-related patterns in the hippocampus, there's a hypothesis. I'd say that there's some evidence for this hypothesis that one of the ways that the hippocampus is able to very rapidly form memories of experience is because that there are pre-encoded sequential structures. And so all it has to do is sort of pull a pre-encoded sequence out of its library of sequences and then paste on the details of the event onto that pre-encoded structure. I think that that's a very elegant idea. We did not see any evidence of that, and we did look a little bit. Cameron is very interested in pre-play, and so we spent a lot of time looking for it. And it seems like it's to the extent that that happens, the sequences are not reliable enough to produce the structure that would be consistent with the hidden Markov model. So it seems like that those sequences must be very random, which I think also from a mechanistic perspective is super interesting. How is it that you're able to produce many different random sequences? You know, it's
Okay, thank you very much. It's been a super nice talk. Thank you. Okay, so Vitor, it's your turn.
Can you hear me well? Yes? Okay. And you can see my screen as well. Okay, good. So thanks a lot, Andrea, for the invitation, and thanks everyone for joining. I'll try to be quick. I'll try to finish in 20 minutes. I'm not tenure, so maybe I can do it. So I'm going to present some results we had in a paper published in 2018. And because of the nature of the symposium, I'm going to try to focus on the analysis part of the work, but I'll give you some very quick introduction on the background so you have some context of the study. Like Caleb made my life very easy because he already explained a lot of the things I had to explain. So yeah, I'm interested in the hippocampus, and as he said, is a brain structure that's very important for memory. And I'm specifically interested in this region for this project, at least. So it's C1 field in the hippocampus. And more specifically, even I'm more interested in what are the functions of these two inputs to C1. So the input coming from C3 and the input coming from internal cortex. So as Caleb showed, when you record data from hippocampus, in this case from C1 specifically, from rodents during active behavior, you have these very prominent oscillations that have around 8 hertz, which we call theta oscillations. And a very strong characteristic of these oscillations is that they have these nested gamma oscillations within them. So for example, here in this theta cycle, you can see my mouse pointer? Okay. So in this theta cycle here, for example, you see that in this theta wave, you have this burst of gamma activity surfing in the wave. And in fact, if you compute the instantaneous amplitude of different frequencies within the gamma range as function of theta phase, you see that naturally you're going to divide the gamma like broad range in 3 different bands, which I'm calling slow, mid, and fast gamma here. So lots of work have been done in the last at least 25 years to review what are the underlying bases of these oscillations. So what's the mechanism that generates these oscillations? And in fact, we now know they have, each of them have like an associated circuit in the hippocampus, which I'm going to try to summarize very quickly in this cartoon. So this black trace here represents a theta cycle as recorded in the pyramidal layer of C1. And we know that when we are at the peak of that cycle, you have most of the spikes from the entorhinal cortex, layer 3, which projects to C1. At the same time, you have the emergence of these mid-gamma oscillations, which you can trace back to stratolacunoso molecular layer, which is precisely where C1 receives lots of inputs from entorhinal cortex. So this evidence, together with more evidence that I don't have time to go through, really indicate to us that these mid-gamma oscillations are reflecting the projection from entorhinal cortex to C1. A bit later in the cycle, in the descending phase, you have the emergence of these slow gamma oscillations with a very similar story. We have a lot of indications that this oscillation is reflecting the inputs from C3 to C1. And finally, later in the theta cycle, you have the activity in C1 being more expressed. And then at the same time, you have these fast gamma oscillations, generally locally generated within the pyramidal layer of C1. So basically, we have three events that seem to be temporally coordinated along a theta cycle. So first, you have the inputs coming from entorhinal cortex, reflected in these mid-gamma oscillations. Just after, you have the inputs coming from C3, reflected in these low gamma oscillations. Then you have like an integration of these two inputs in the dendritic trees of C1, and then C1 generates an output in the trough of the theta cycle, which is reflected by the fast gamma oscillations. So just let me go one step back and show you a very basic analysis on how we quantify this gamma theta coupling. So we start with the raw LFP, which I'm showing you here in black, and then you have the theta component, showing here in red. Then you find some time frequency representation of the frequencies above theta. And then you just need to correlate the instantaneous amplitude of each of these frequencies to the ongoing phase of theta. And if you do that for as many cycles as you can get, you will find our output like this one. So you see that you have an increase in power in the mid-gamma range just after the theta peak, and you have then just later an increase in power in this low gamma range. If I was showing you the faster frequencies here, you would see a fast gamma there as well. Okay, so what I really want to call your attention to in this figure is, if you look to the individual cycles I'm showing on the left, you're going to have a very hard time to see this average cycle really expressed in real life. So basically, this average cycle is not really representative of the individual cycle. So for example, this cycle here seems to be mostly dominated by mid-gamma oscillations, as the next cycle is mainly dominated by the mid-gamma oscillations. I said mid before, I meant this. So one hypothesis that this is just biological noise, so it doesn't have really a meaning. Like it's like the systems are complex and generates these fluctuations, very hard to but yeah, they don't give us any information. But the other hypothesis is that we could extract information from this variability and really try to infer what the brain is doing. So if you really believe this low gamma is reflecting C3 to C1 inputs and mid gammas are reflecting entorhinal cortex to C1 inputs, I could conclude or infer that in this cycle here, for example, which is dominated by slow gamma, C1 is reading more strongly from the information coming from C3. Whereas in the next two cycles, because I have more mid gamma, I'll be like C1 will be reading more strongly from the entorhinal cortex. So this is what we want to explore in this work. So I'm just going to show you first how I'm characterizing this cycle by cycle variability. So I start with the raw LFP again. I find a theta component. So I take out the theta component and the slower components from the data. So I have the supra theta components. Then I use wavelet analysis. So just compute a wavelet spectrogram to get the time frequency representation of the supra theta components. And then, because I'm interested in analyzing cycle by cycle information, I'm detecting this single cycles of theta, as I'm showing here in these dashed lines. And what I decided to do next was to average these windows for each theta cycle. So this white trace that you see here is the average of the spectrogram within this window. So it's like a power spectrum, let's say, of each cycle. So basically, they give me the mean amplitude of a given frequency at a given cycle. I'm calling these spectral signatures. So if I average all these traces, I'll get something like this. So yeah, high power in the gamma range, but you see that in the shaded area, I have a very large standard deviation. So I have lots of variability in the data. So the next step we are taking is, okay, we're going to apply a pattern recognition algorithm to this data set because we want to extract like reoccurring spectral patterns across cycles. And we decided to do that using ICA in this case. So I'm going to go through the independent components I find when I apply ICA to this data set, and I just try to understand each of them. So I get one principal component, one independent component, which I'm calling here theta spectral component, so I remember what they really represent, which is attributing high weights to frequencies above 100 Hertz. So this seems to be extracting the fast gamma oscillations. I have another one here, which is attributing high weights to frequencies around 35 Hertz, which seem to be extracting slow gamma oscillations. What I'm showing you here in this square is just a sanity check. So I'm basically doing the same analysis as I did before, but instead of using all cycles to compute this plot, I'm just using the cycles that were strongly expressed in this component. So it's just to show you that I can get a subset of cycles that were mainly dominated by these low gamma oscillations. Then I have two independent components in the mid gamma range, it's in the story. And then I get another one that was more unexpected, which is giving me high weights to frequencies around 22 Hertz. When I check the cycles that have this component very strongly expressed, I see that they have this 22 like beta component peaking in amplitude near the peak of the theta cycle. So different phase as the slow gamma. Okay, so just to I really like to go back to the raw data and see what I'm doing in every step I take. So here I'm just showing you single cycle examples, the raw data of single cycle examples for each of these groups I showed you before. So just going from the right to the left, this one on the right was a cycle that had this TSC4, which is the 80 Hertz gamma component. You see that in fact you have this the theta cycle here, and on the top of the theta cycle near the peak you have this gamma burst, which is around 80 Hertz. Very similar story for the 50 Hertz component. Here I have the slow gamma, so see that again I have the theta cycle here, and then the slow gamma burst closer to the trough now. And the TSC1 that I was calling like a beta. For now, it doesn't look really like an oscillation, but it seems that you have the theta cycle, and just after the theta peak, you have this negative deflection on the LFP. So it's like a, let's call it a beta dip, just after the peak of theta. And here I'm just showing you, so all these examples are coming from the same electrode in the same session. Okay, so it's just that the signal, whatever happens in that theta in the cycles, that electrode are changing dynamically. So these are just more examples of these TSC1 cycles, just so that this reoccurs in the data quite a lot. So we have this dip in the LFP. These are more for the slow gamma, just more like raw data examples. You have the theta cycle and the slow gamma riding that wave. And then the same for the others, but I'll just go quickly here. Okay. Before I mislead you to an interpretation, I just want to make very clear that I'm not claiming there are clusters of theta cycles in the data. So I'm just plotting this scatter plot here, in which each point here is a theta cycle, and just plotting them with respect to two components, just a random example. And see that there's no evidence of clustering. So I'm just saying the theta cycles in terms of the spectral components can go one way or to the other in the axis, but they don't form clusters. It's just like they lie on the continuum. Okay. Okay. What's very convenient is that if I apply that same like semi-unsupervised framework to different animals or even to different species, so I tried in mice and rats, I get very, very similar spectral components. So it's very, very robust. Given I'm recording from the pyramidal layer of C1 during active behavior. So it wouldn't work the same way if I'm analyzing REM, for reasons I don't have time to explain now. Or obviously, if you go to other brain region, you're going to get different components. But it's very, very robust if you are in C1 during active behavior. Okay, so far I just showed you that I can get these components from the data, and okay, it's nice that they are robust, but I didn't show you any evidence they are meaningful in any way. So I want to validate them somehow. So the first obvious thing I can do is to see if I find a correlate of these components in the spiking data. So what I'm showing you here is the average firing rate of principal cells in C1 around the peak of the theta cycles. So here I'm showing you this classical result in which you have an increasing rate of principal cells close to the trough of the theta cycles. Now I can do exactly the same analysis, but instead of using all theta cycles I have, I can just do it for a subset of cycles that were dominated by a specific gamma component. And when I do that, I see some deviations from this average. So for example, if you have strong 80 gamma expressed in the theta cycle, you tend to increase the rate of the spikes, specifically at the peak of the theta cycle. But if you have strong slow gamma, you increase the rate at the trough of the theta cycle. So just plotting this way, because it's easier to see, this is just a relative change to the average cycle. So see that there are different spike temporal pattern bias expressed in the pyramidal layer, depending on which gamma component you have in that cycle. Next, I checked if the correlation between these gamma components and the spikes are also present in a finer time scale. And I really like to use spike trigger averages or trigger averages in general to do that. So what I'm doing here first is in this black trace here is the mean LFP, so the mean raw LFP that I get if I average them across these beta deeps that I showed you before. So it's like a beta trough trigger average of the raw LFP. This is the waveform I get. But what's really important here is what happens to the spikes. So this raster that I'm showing here, so each row in this raster is a different theta cycle, and each tick is a spike of any principal component I recorded in that day. So it's like a multi-unit of all principal components. So see that just by aligning the cycles by this beta deep, I have clearly seen these stripes of inhibition, excitation, inhibition in the spikes. And obviously, if I do a mean PSTH of these cycles, I get something like this. So for free, I see the reflection of the beta component on the PSTH. And all I had to do was to get the one beta trough per theta cycle and aligning the activity with that reference. If I do the same for the slow gamma, I get a very similar result, but now for the slow gamma scale. So I just want to emphasize a lot, something that for me is very important, is that I only get, in this case here for the slow gamma, one trough of slow gamma per theta cycle. Align the activity of the theta cycles, and then I get 3 cycles in the PSTH, showing you clearly that around that slow gamma trough, you have an oscillation of the rate of the spikes, matching the slow gamma pace. And even more striking, if I do that for 80 hertz gamma, even I see the same, which for me is not really clear how, because remember this mid gamma is arriving in the apical dendrites, so it's very far from the soma, and still they are managed to create this oscillation in the output of the principal cell layer. So we can hypothesize that it's another interneuron there that receives and propagates that oscillation, but I don't have any evidence for that right now. So in general, all spikes we had are firing at the trough of these gamma components. So they fire at the trough of the theta cycles and also at the trough of the gamma nested components. Okay, so I showed you that, okay, the spikes seems to be, there's some kind of co-modulation between the spikes and these gamma components, but then I finally can go to my ultimate goal, which is I want to use these gamma components to study the function of these projections they are reflecting. So I'm going to show you one example on how we can do that. So I'm going to do a reactivation analysis, which is similar to what Caleb showed, but I'm more concerned about co-activations here, not the sequences. So the way it happens is we let the animal sleep. So we get a baseline sleep. And instead of, as Caleb showed, instead of theta, we get these repo events. And we want to see which cells are co-firing across these repo events. So in the way we are doing to measure that is by means of a correlation matrix, very simple correlation matrix. So basically we have the correlation of a pair of cells, or every pair of cells, across repo events. Then we let the animal explore an open field, and then we do exactly the same analysis, but instead of using the repo events, we use now theta oscillations. So theta cycles as the window. And then the animals go to sleep again, and we do exactly the same analysis. So we say you had a reactivation of the coactivity patterns expressed in exploration if the correlation matrix expressed during exploration is more similar to the correlation matrix expressed in the post-sleep than it is to the pre-sleep. And the most straightforward to compute that is to use a partial correlation or linear regression. So you compute the similarity between the exploration correlation matrix to the post-sleep correlation matrix, regressing out what you had before exploration. Okay, but what I can do different now is instead of using all cycles in the exploration, I can just go for a subset of cycles that I can choose based on which gamma components are there. So here I'm comparing reactivation level I get for cycles that are dominated by slow gamma or mid gamma or any of these gamma components to cycles I sample at random. Okay? And you see that in this case here, the choosing the cycles by the gamma components is no different than just randomly sampling the theta cycles. The thing is here, I was doing it for a familiar environment. So the animals were exploring an environment they knew from before for many explorations. If I do the same thing, but now the animals were exploring novel environments, so the environment they've never seen before, suddenly I have these two reactivation levels jumping out of the random distribution, and these are precisely the mid-gamma, these theta cycles dominated by mid-gamma oscillations. So what this is showing is that, in, let's say in a mathematical terms, more mathematical terms, is that these theta cycles that had strong mid-gamma spectral components are more reactivated in post-sleep. And in other words, what we're trying to infer here is that the coactivations that happen in C1 when entorhinal cortex was more strongly dominating or driving C1 are the ones that are being selected to be more reactivated in post-sleep, indicating that the novel information that really needs to be consolidated is more coming from the entorhinal cortex during the exploration. This is just a complementary analysis showing you the same thing, but I'm going to skip just because of time. And then I'm just going to summarize the talk. So I'll show you that we can use ICA to extract the spectral components that are consistent across animals or even species. These spectral components associated with different spike temporal patterns. This is one result I haven't shown because of time, but each of these components are also associated or correlated to a distinct cell assembly in C1. Theta cycles that are dominated by mid-gamma are more predictive of what's going to happen in post-sleep sharp wave repos. And also a result I haven't shown that these nested gamma oscillations are differently related to different stages of a memory task. So depending if in the memory task, the animal is performing more like a retrieval or an encoding task, these components will be different as well. So just want to thank you everyone that was part of the work from the pre-lab. Ben for helping me to design this cover, and to Abilasha, Mohammed, and Helen for the inputs during the course of the study. And yeah, happy to take questions.
Thank you very much, Vitor. Is there any question? I think I have, well, I have a quick question for the maybe attendees that are not much into neuroscience. Can you give an intuition on why characterizing inputs and timings of brain oscillations is basically essential for brain computation? Like more, yeah.
Okay, so it's a very good question, but I'll try to. I'm not really an enthusiast of, so it's a very personal answer, okay? I'm not really enthusiast of the function of oscillations per se, or as oscillations. The way I think these oscillations are very, very interesting because they are proxies for something that's happening in the brain. So for example, I'm quite confident, for example, that slow gamma in C1 reflects this projection of C3 to C1. So, and I'm very interested because we really want to understand why, like we want to understand the whole hippocampal circuit, and this is a very important input, a very important projection within the hippocampal circuit. So, and it's very hard to really observe this projection directly. So I'm basically using this slow gamma to infer this projection. And this is how I like to use oscillation. So yeah, there's a lot of very elegant theories saying that all this can provoke a resonance or a filtering of information. And I'm not really enthusiastic of that, maybe because I don't know the right papers to convince me this exists in the brain. But yeah, the real importance for me is this kind of thing. They are really proxies, so we can infer what the brain is doing, as much as you would have in particle acceleration. You don't observe the collision of the particles themselves, but you see what they provoke. So it's very basic principle, very similar principle.
Okay, thank you. We have another question. It's in the chat. Well, Vitor, thanks for the presentation. Ruben Rodriguez says, do you think the better understanding of the human brain could help us to design better models in RIIAA, artificial intelligence areas?
I'm not an expert on that specifically. I know I have a very good friend that works on biological inspired models for AI applications. And I know this is starting to be very trendy and very useful, but I really don't know much. Like mine, I think I'm very pessimistic in general, and I would have the reflex to say no, because AI is very different from the solution. The things that we want to solve in AI are very different from the evolution course of the brain. But I more and more see people giving me examples of how doing like biological inspired models are helping to do applications. So yeah, but I'm really not an expert. I'm not a good person to answer that question, I think.
Yeah, you have answered yes. So I guess it depends. Okay, so thank you very much. I think I'm going to need an extra 10 minutes, if that's okay.
Yes, that's fine.
Thank you. Let me check. Okay, so I'm going to talk about how we use convolutional neural networks to explore electrophysiological events. I think with the talks before, I can also more or less skip this part. I just wanted to to have a little review about why I do think that brain oscillations are very important, because here you can see a graph. There are many rhythms in the brain, for example, the heart rhythm that changes with the size of the animal. So mice has a high frequency heart rhythm, but I don't know. Us, for example, humans have lower. However, brain oscillations are really preserved along it. They are independent on the brain of the size of the brain. So this means that something about the frequencies and the brain's oscillations are essential to neural computation. Each of them has different functions, and I'm going to talk about ripples. That is something that both Caleb and Vitor has talked about. They are essential for memory consolidation. And well, in fact, something that also Caleb tells, if we can study memory by detecting and intervening, like stopping this from happening to study memory. Here you can see like the brain signals, and these in green are the ripples that are high frequency events, basically. Okay, so the standard methods usually rely, as these are frequency events, they rely on thresholding a filtered signal. But as I am very, very interested in brain oscillations, I think that the shape of the oscillation can tell us about something about the neuroscience, the encoding behind. So this is something that I think we should try to solve, and also that the events that we are going to detect, they are biased toward high amplitude ripples. So our goal is to use machine learning tools to detect ripples online, to intervene, and offline to do analysis, and also explore why the CNN converges to the solution it takes. Yeah, so why convolutional neural networks? Well, as I suppose many of you know, they are the state of art of many of our daily tools. For example, they are used with face recognition, autonomous driving, text classification, and they require very little preprocessing, so they are ideal to use them in an online context. By the way, yeah, I'm also talking about like these ripples also occur in the hippocampal part. So yeah, we are very hippocampal people. So what we are going to do is we are going to train a CNN whose input is going to be this brain signal. It's going to be a one-dimensional convolutional neural network that is not so common. The two-dimensional are the common ones. These are the inputs. They are going to be 8 channels, and each channel is going to be like a color in the imaging equivalent. And we are going to feed the data in 32 millisecond windows. This is for the sake of the online context. And the output is going to be a probability between 0 and 1 of these 32 millisecond windows having a ripple. Okay, so this is the architecture. The design was like a modular design. The input is going to go to the first module. Each module is going to consist on 3 layers. The first one, a convolutional layer, and then a batch normalization and liquid value layers. The convolutional layer is the one which is in charge of the processing. And the processing is made basically by the kernels. The kernels are matrices of weights. The height corresponds to the input, and each of these 8 channels is going to go to each of these rows, and this is the temporal axis. So basically, what the kernels do is that they perform a convolution that is basically a pairwise multiplication from these weights with the data, and they output a number. So during the training, these weights are changing until each kernel is specialized in detecting a particular feature. So the output of the convolution between one kernel and the data is going to give like an idea of how present this learned feature, this encoded feature, is in the data. And well, studying the kernels later, we will see how this can, what this can tell us about the ripples. Here you can see the extended architecture. Well, finally, we add a dense layer, which has a sigmoid function. So the output is a number between 0 and 1, which is the probability of being a ripple in there. Here is the extended version of the architecture. Here, for example, this is the first layer. It has 4 kernels, and each of the outputs goes to the batch normalization and the RIIAA layers that provide stability along the different layers. And then the output of this is fed into the next convolutional layer, and then again 7 of these modules up to the last the dense layer. So here are the results. We are going to measure the performance with 3 numbers: the precision, that is the number of hits over the total number of predictions; the recall, that is the number of hits, but over the number of tags, of true events labeled by an expert; and the F1, that is the harmonic mean between these two. So this is the best way to measure the performance of the network. Here I have to tell you one thing, that unlike the imaging world in convolutional neural networks, for imaging, you are, for example, you want to classify if an animal is a cat or a dog, and we can label, and by 100%, every people in the world is going to label the same if this is a dog or this is a cat. Well, this does not happen with neural data, because if you give these 2 different experts the same data, the tags are going to be partially different, because it's not as clear as with, I don't know, images. So here is why I have this like gray region that I'm going to call experts region, because what I did to evaluate how good or bad was these performances was to give the same data to 2 different experts. They tagged the data in 2 different ways, and then I analyzed what was the performance of one expert versus the other expert, and this was the result, this gray area. So as you see, the convolutional neural network lays within so the performance is it's the same as the expert. So, and this is the same with the filters. They both lay within this expert area. However, the good thing about the convolutional neural network is that it seems to be more consistent, maybe a bit low, but more consistent, which is going to be good for something that I'm going to talk about a bit later. The good thing, though, is that when we did another measure that was like to take into account to measure the anticipation, like when in an online context, when we did detect the ripple, if you compare the standard method, the filter, and the convolutional neural network, it seems to be that the convolutional neural network sees something in the data before it happens. So it sees the ripple before the filter. Well, and that itself is pretty cool. But we wanted to, like, our interest was also like how exploring what's the CNN solution. And for that, we went to the kernels. We know that the kernels perform a convolution with the data, and this gives an output which finally, after many layers, is going to be the probability of a ripple. So we wanted to explore the weights of the kernels to see if we could infer what the CNN was looking at. What were these learned features? Here you can see this is the kernels of the first layer. So this is like this length is equivalent to 4 milliseconds in the data. That is more or less this. So here are the kernels after training, the weights of the kernels after training. Let's focus, for example, on the first one. Here you can see that there is like a gradient along the channels. So this seems that it's going to be activated when the data of the upper channels go into one direction and the data of the lower channels is going to the opposite direction. So do something like going up and down, or vice versa, down and up, something like this picture in here. However, the second kernel, or more or less the other one, seems to do like more or less the opposite. Instead of focusing on the on this, I don't know, like expanding and shrinking, it's focusing on a horizontal gradient. So this is going to be, if this is 4 milliseconds, it's going to look for things that go from down to up, like 2 milliseconds down and then up. So I would guess that, for example, if we see this in here, this is going up and this is going down. So this kernel would activate in this, like during this 4 millisecond 4 millisecond window, and then it's going to deactivate in the next 4 millisecond window. Well, as here it does the opposite because it inverts. And in fact, if we see this, these are the kernel activation, the output of this convolution of these kernels. We see this, like the first kernel is like capturing a global behavior of this ripple going, like doing this expanding thing, and yeah, it gets this much more global behavior. And these kernels that were focusing on the local going up and down, they tend to capture in fact, this frequency event, no? This oscillation, this frequency oscillation patterns. Okay, this is super nice, but we cannot do this with the rest of the kernels because deeper layers are very, very much more difficult to interpret. So we did another thing, and this is the last thing I'm going to talk about. We know from images that there's another way to look at how, well, here I tell how a ripple looks like inside the CNN's head. And this is translated into a more objective question, that is, what input would activate each kernel the most? And for images, we already know that the first layer look for repetitive patterns like edges and textures like this. And the last layers looked for more complex things. For example, this is the preferred input for a unit in a convolutional neural network that which output is the probability of being a banana. So here you can see this preferred input is like, I don't know, like many bananas overlap one over each other. I don't know, I think it's pretty cool. Here you can see the starfish, and I said, okay, let's do the same. What is this, but for a ripple? The way that we have to do that is to apply a gradient ascent over the input space. So basically we take a random image of noise, and then we update this input image in a way that the kernel activation is higher, is the highest, and that's why this is gradient ascent. So doing this in an iterative way, we achieve the overlap banana image for this unit in particular. What happens if we apply this method to our, like the dense layer, the last layer of our network? Then I was expecting, like this super nice, I don't know, ripple thing, because as I said to you before, there is not a global way of defining a ripple. So I was expecting, like this is going to solve our problems. And then I had this that is like looks kind of horrible. This, I wouldn't say this is a ripple, but in fact, I mean, I repeated several times and the solution was always the same. So I repeat, this is the input that activates the last layer the most. And in fact, if you do a correlation between this, I don't know, yeah, this pattern and 4 different categories of data that are like the true positive, that are the predictions that coincide with the ground truth, the false positive, that are the predictions that were not in the ground truth, also the false negatives, that are the missed ground truth, so the network didn't predict these events, and the true negatives, that are the events that both the network and the ground truth say, okay, these are not events. So if we do this, we do the correlation between this pattern and these events, this group of events, we can see that obviously the highest correlation is among the hits, but that there are these events that weren't in the ground truth, but the network say, okay, these are ripples, these are more similar to this than the missing ground truth. And if we see examples of this group, we can see that actually some of them were sharp-wave ripples, which is super cool. Some of them obviously weren't, because here you have an example of an artifact, and brain signals have a lot of artifacts, and that's one other problem that we have to solve, how to avoid detection of this. And again, there were some events that some experts say they were ripples, and some other experts say that they weren't ripples. So for me, this was a nice thing, because it somehow came up with the idea of generalization, like, okay, maybe the network is looking for something that they are like actually ripples, and for that we can try to, I don't know, somehow interpret again what a ripple is, what this is brain's oscillation, but just in terms of the oscillations per se. And we did the last thing that we tried to see this generalization. So we had this one network trend, and then we computed the F1 with the 2 different tags, ground truths, with the expert that tagged the data which was trained with, and another different expert. And here you have the results of the performance of with the original, the ground truth of the original expert. What's the performance with another ground truth, a different ground truth, and you can see that these are different. And finally, what was the performance if you combine both tags? And as you can see, there is a significant increase in the performance, which gives the intuition that this network is somehow generalizing what the concept of these ripples is. And we did the same with the filter, and even though there seems to be also a trending into going up, the dispersion is so big that it's not significant. So the conclusions are that using convolutional neural networks, mid-dimensional convolutional neural networks, we have been able to detect ripples before standard methods, to interpret what a ripple is by studying its learned features, and generalize a partial ground truth. So, well, thank you. I want to thank my lab, especially, well, Rodrigo Amaducci, that is co-author of this work, and also my PI, that she's an excellent scientist, and I love her very much. And of course, thank you for attending. So if you have any question, I'm sorry, I'll try to be quick.
I actually have one question, and I can take advantage of my position as an organizer. And it applies for any of the speakers, I think, could answer this, which is, do you think that if we had like a much better resolution, like if we come up with neuroengineering tools that allow you to record time-wise and location-wise with much better resolution, like let's say neural level resolution at all times, would it then be easy to understand what's happening and how the brain computes, or would there still be like a lot of effort needed in terms of like analysis of that data to understand the algorithm of the brain?
Well, I think that would be amazing. I hope someday we can have that, but still I think there would be like tons of effort, because obviously tons of data would help, but I don't think we have much clue to next day have the solution, you know. I don't know what you guys think.
I think that compute is a complicated question. I think that if the question was, you know, how does a brain detect bananas? I love that slide, right? So how does our visual cortex identify bananas, among other things? My guess is that having more data would allow us to become a little bit more, have a clearer understanding of that. But already our artificial intelligence and machine learning tools have given us some really strong hypotheses how something like that might work. For more complicated things, which are the things that we want, like how do we drive a car? I think that the challenge is much larger, because you need to, just having the data would not necessarily help us, I think, understand that a good experiment is required to get good data.
Yeah, I think more data is always welcome, of course, but we are ready, like, and it would help, yes, but we are already struggling sometimes to analyze the data we have now. So in some recordings that we have hundreds of neurons, people are not going to agree what, you know, we also need, I think we are a bit behind on the methods as well, on the statistical methods to deal with a very high dimensionality and not long recordings, basically. So yeah, so I guess it would improve, because we could do some stuff already if we had more data, more localized, more detailed. But I think the big bottleneck now is to develop statistical tools to deal with the data.
I actually want to ask Andrea a question, but it relates also to your question. So if you had, do you think, when we've looked at ripple detection, false positives are always a challenge. And we've imagined that having other electrodes and outside the hippocampus would be helpful. And so have you thought about that? It seems like that's something that would be valuable. And obviously, I think in answer to Andres' question, sometimes one more electrode in the right place might be awesome. So if we had 10 million, then one of them might actually be the one that we want.
Yeah. In fact, this is something that we are now doing with, well, because there, like in the last year, there has been amazing advantages in the technology. For example, we have something to register from many regions in the brain, and that's like amazing. And we are planning to use this for the NeuroPixels probe to see what's happening along different regions. We were interested in like, for example, knowing what's basically in the hippocampus, but in like in regions that precede, in the information flow that precedes the CA1 region. But yeah, we are still doing that. We are closing this, and the next step is of course like seeing what's happening in other brain regions just before or at the same time, or maybe after the ripple occurrence.
Seems to me, Andrea, that what you need is some neuron or some system that can identify the ripples for you. So then you will solve the problem of the tagging, the spread tagging, because those neurons will do that for you. So in fact, it's relate the ripples to the function. This is what you need. Some examples of it. I have a question regarding your analysis. How how important do you think is the amplitude of the signals in the ripples versus the timing of the different events that compose the ripple? Because the amplitude clearly depends on the place where you have the electrode. The amplitude, the same ripple in one place can be very different in amplitude from the same ripple recorded in another place. So how important do you think is the amplitude? Don't you think that maybe the spatial information, the specific waveform of the ripple can confuse a little bit the convolutional neural network?
So for us, the amplitude was important in terms of because we, like when we were visualizing the data, there weren't like, so the first thing is to say, okay, the ripples are an 0-1 event. So there is not a ripple, or there is a ripple, or there is a continuum. And for us, like looking at the data, we thought that, yeah, that there were some events that were, so these events that are not clear if they are ripples or not, but they have a structure in the spiking. So that I think those are events that are, so they have low amplitude, and it's not dependent on the distance of the electrodes. So what you say, it's true, no? Like if you have a ripple very far away, the amplitude is going to be lower. But we were thinking more in those, I don't know, events that are emerging to be a ripple, and that's what I think we should try to look at, because I don't know, it's a regime in which there has not been a lot of exploration. Like what happens? What's this event that is emerging?
I have a very, very quick question to Andrea. I missed how you defined the ground truth.
So basically, we gave the experts the data, and and they went through it, like, like second by second, and they tagged, and that's it. Yeah. So.
But then, how you compare the ground truth to the expert performance?
So we give the same data to two different experts, and they tagged ripples, and we did as we, for example, to compute the performance of the network, we compare the predictions with the ground truth. So to compare the experts, we say, okay, expert one is the prediction, and expert 2 is the ground truth. What's the performance, and what happens if you do the opposite?
And when you compare, and you compare to the network, you also choose one of the experts, or
yeah, we
get a
did
consensus?
two different things. For the first thing is to compare experts between themselves, and the result was like 70% of performance. So this clearly tells you how ripples are not something that we can define so easily. And we also did, we also compared what was the performance of the network with one expert, with the ground truth of one expert and the ground truth of the second expert.
Sorry, I don't know when we'll get kicked off, but I actually have another question. I think this is something, the idea of predicting ripples is something that in my lab, we've been very excited. We've thought a lot about. And I always imagined an LSTM would be a better architecture than a CNN for that problem. And so I'm sure that you've thought about this. I'm just curious about what challenges you might have faced trying to implement an LSTM as opposed to CNN approach.
Yeah, in fact, so we haven't focused, we first wanted to detect them and then predict them. So yeah, next step is to predict them, but we did some analysis with LSTM, and actually the results weren't so good. I don't know, for us was also something surprising, but when we saw that the CNN performed better, we just kept with it. We didn't go much into, we didn't do much, but we tried it. Maybe for prediction it's better, I don't know, but
All right. I don't know if there are any other questions, but if not, I would like to thank you all. It was amazing. It's an honor and a privilege to be able to have such a course and then a conversation like that. I thank all the attendees too. For the attendees, I would like you just to fill the form if you haven't. And for the speakers, I saw Vitor and Caleb share their slides. Feel free, Andrea and Pablo, to share your screens. Just put them there. And thank you. Thanks a lot. This was a great beginning. Andrea, if you can make me a host, because for some reason I got disconnected for a second and I'm not a host anymore, so that I can stop the recording. But otherwise, thank you, everyone.
Thank you for having us. We really appreciate it. Thank you, Andrea, for organizing.
Yeah,
thank you
thank
very
you.
much.
Thank you. See you next time.
Yeah. Bye-bye.
Bye.